Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss pyroptosis. Pyro in pyroptosis in Greek stands for fire or fever, while tosis refers to falling. So that tells us that this programmed cell that is going to lead to fever. This is a programmed cell that is induced by the formation of gasdermin. It's a protein, gasdermin pores, that gasdermin holes in the plasma membrane, thereby killing the cell. Now, pyroptosis is very predominant in cells of the innate immune system, such as macrophages, dendritic cells, and so on. However, pyroptosis has also been observed in non-immune cells, such as the epithelial cells lining the airways or the intestinal cavity, neurons, as well as the vascular endothelial cells. So as in previous programmed cell deaths, pyroptosis starts with some signal binding some sort of a receptor. In pyroptosis, there are several different kinds of signals. One class that we call PAMPs, pathogen-associated molecular patterns, and the other class we call DAMPs, which are damage-associated molecular patterns. So PAMPs come from pathogens. DAMPs come from broken cells, the host broken cells. Now the receptor for these PAMPs or DAMPs are called pattern recognition receptors, or PRRs, or we can call them sensor proteins. And there are several different types of PRRs. Examples include toll-like receptors, C-type lactin receptors, nucleotide binding domain and leucine-rich repeat containing receptors, or NRRs, AIM-like receptors, ARRs, RIG1-like helicase receptors, as well as XLRS. So many different types of PRRs. Why? Because there are many different types of signals. In this video, we'll look at a few examples of PAMPs and their respective pattern recognition receptor. So we'll first look at the PAMP called lipopolysaccharide, or LPS. This one doesn't have any receptor, so we'll look at this first. Then we'll look at the other types of signals, including proteases, various types of proteases that have their receptors, toxins with the respective receptors, double-stranded DNA, which is receptor AIM2, proteins of flagellin, rod or needle proteins found in bacteria whose receptors are various NAIPS, and then we have signals such as toxins as well as danger-associated molecular patterns and crystals such as cholesterol and uric acid, and they have their own receptors such as NLRP3. So let's first see how the signal LPS, this PAMP called LPS, lipopolysaccharide, causes pyroptosis. So the LPS that is outside the cell could enter the cell by various ways. It could enter the cell by bacterial vesicles. The cell can take this up by endocytosis. Or the cell could have proteins these are host proteins that can act as chaperones. And these chaperones can bring the LPS into the cell. Now, once LPS enters the cell, it doesn't need a receptor. It will directly activate procaspase 4 or 5 into active caspase 4 or 5. So no receptor necessary. The activated caspase 4 or 5 will then break down this protein called gasdermin D protein, or GSD-MD, into the N-terminal fragment, or NGS-DMD. Now this NGS-DMD will then oligomerize, will self-assemble and insert into the plasma membrane, forms these pores or these holes in the plasma membrane. This formation of holes make the plasma membrane permeable, and the small cellular contents from within the cell, small DAMPs, can start coming out of the cell. And of course, these DAMPs can attract immune cells, activate immune cells, trigger inflammation. And as before, the permeabilization of this membrane leads to the entry of sodium as well as water, eventually causing cell swelling and so on, and that triggers the rupture of the plasma membrane rupture, PMR. And that can happen through a protein called ninjurin-1 or NIJ1. This protein basically forms filaments, and these filaments essentially cut the membranes apart, just basically destroys the membrane. It makes huge holes in the membrane, and that's going to lead to the loss of even very large molecules, including very large DAMPs. And of course, once the plasma membrane is broken, filled with holes, the cell is going to die. So that's how the lipopolysaccharide leads to cell death. Now let's see how the signal proteases, certain enzymes that break down proteins that come from pathogens, how do these proteases cause pyroptosis? So in this case, notice there is a receptor for some of these proteases. So what happens is these proteases will bind pattern recognition receptors called NLRP1, which stands for nucleotide binding oligomerization domain, leucine-rich domain, and pyrene domain containing 1. So NLRP1. And this binding is going to cause this NLRP1 to oligomerize and form a sort of a scaffold in the cell. Now this NLRP1 has a PYD domain, and this PYD domain will interact with the PYD domain of another protein called ASC, apoptosis associated speck-like protein. And this ASC will now be recruited towards the receptor. The recruitment of the ASC to the receptor will now form what we call the inflammasome. Now, besides the PYD domain, ASC also has a card domain, which we also see in caspase-1. Caspase-1 also have the card domain. So through card-card interactions, ASC will recruit caspase-1, and it will activate the pro-caspase-1 into active caspase-1. This active caspase-1 will now cleave and activate the cytokine IL-18. So now we have this activated cytokine IL-18, and also the pyrogen. Pyrogen is a chemical that causes fever, called IL-1 beta. So this pro-IL-1 beta inactive form becomes an active IL-1 beta. And that's going to, this release of these things here is going to lead to inflammation as well as fever. Now the activated caspase-1 is also going to cleave gasdermin D into the N-terminal fragment, NGS-DMD. So we see this N-terminal fragment being formed. And this fragment will oligomerize to embed into the plasma membrane and will form holes or pores in the plasma membrane. The plasma membrane integrity is therefore disrupted, and small DAMPs and also the pyrogen IL-1 beta, which is also small enough that these will be released by the cell. And these DAMPs, of course, will trigger inflammation, while this pyrogen will cause fever. The increased permeability of the membrane will once again cause sodium and water to enter the cell, and eventually the cell plasma membrane will be broken up into huge, there'll be huge holes through this protein ninjurin 1, which forms filaments and basically break apart the plasma membrane, as we talked about earlier. And now with these huge holes, with this huge disrupted plasma membrane, very large molecules are going to get out. That includes these DAMPs, huge DAMPs, and that will now again cause even more inflammation. Eventually, the disruption, the breaking of the plasma membrane is going to cause the cell to die. So with these two examples, now we should be able to see what the rest of the signals do, such as some of the other proteases, toxins, double-stranded DNA, and so on. So let's talk a little bit about how the other signals cause pyroptosis. So some proteases will bind to receptor called CARD8. So when these CARD8, which is caspase activation and recruitment domain 8, when this protein detects and binds the protease PAMPs, this protein becomes activated. And when CARD8 becomes activated, it will directly interact and activate caspase 1 through CARD-CARD interactions. There is no adapter protein that is recruited. So this activated CARD8 will directly bind and activate procaspase 1 into caspase 1. And then this caspase 1 will go and do all these things we talked about, including the formation of these gas-termin pores, and that will cause the cells to release pro-inflammatory DAMPs as well as lead to cell death. What about the signals like certain toxins or double-stranded DNA? The toxins will bind pyrin. Pyrin is adapted from the Greek word fever. Fever in Greek is pyretos. So, for example, pyrexia is a medical term for fever. So pyrin will sense these toxins, while the double-stranded DNA is detected by this PRR called AIM2, which stands for absent in melanoma 2. So double-stranded DNA will bind AIM2, while these toxins will bind pyrin. So this binding will lead to the recruitment of this protein ASE, apoptosis-associated speck-like protein. So ASE will come towards this side, and this will form this complex that we call inflammasome, and that will then cause the rest of the pathway that we talked about, leading to inflammation and cell death. Now, in case of these signals such as the protein flagelline or different proteins found in the rods and needles of bacteria, they will bind these PRRs called NAIPS, which stands for neuronal apoptosis inhibitory proteins. And this binding will recruit this protein NLRC4 which stands for NLR family card domain containing protein 4, and sometimes even ASC is recruited. So with the recruitment of these proteins, we have the inflammasome, and this inflammasome then will cause the activation of caspase-1, and the rest of the pathway is similar to what we talked about earlier. So that's basically the concept here. The PAMP, a certain PAMP will bind a certain receptor, and that may recruit other proteins to form the inflammasome, which then activates caspase-1, and that eventually causes the formation of this fragment, this gasdermin fragment, which then goes and forms pores in the membrane, and the membrane becomes permeable, releases DAMPs, and eventually the cell breaks, leading to inflammation as well as cell death. And that's exactly how these PAMs, such as toxins, as well as DAMPs, such as ATP or crystals, cause cell death. They too will bind certain receptors. So in this case, NLRP3, for example, nucleotide binding oligomerization domain leucine-rich domain, and pyrene domain containing 3, or NLRP3. And that binding will recruit other proteins, for example, ASC, as well as proteins such as NEK7, which stands for never in mitosis A related kinase 7. And this once again forms the inflammasome, which will activate caspase-1, that will then lead to gasdermin cleavage and formation of pores in the membrane, leading to inflammation and cell death. So this is how pyroptosis occurs when the signals such as PAMs as well as DAMPs bind their respective receptors, PRR receptors, and that eventually leads to inflammation and cell death by pyroptosis. In the next video, we'll look at another programmed cell death, and that's called Pyroptosis. So until the next video, once again, thank you very much for watching, supporting this channel, and have a wonderful day.